discussing, not like a very scientific one. So you can stop me in between, and we will start with very basic and go to the advanced application, what we are doing in the agriculture sector, and why this genome sequencing is key to develop any improved variety. So that's why I kept it like the advancement of genome sequencing technology in agriculture. So it's not only the sequencing of plant, but what are the other application involved in the after sequencing? Because everybody is saying that we sequence, what after, what next. So all these things we are going to touch one by one when my presentation will go down. So let's start with that. So I'll start with very basic, that the overview of genomics. So those who are the newcomer, the students who joined, so for them it is very basic knowledge that the genome is first coined by the Hans Winkler in 1920 to define the complete set of genetic information in an organism, including their protoplasm. Right, and the study of genome is known as genomics, and it comprises as the different types of genomes that we will study. And this the genomics terms was coined by the Thomas Roderick in 1986. So this is some basic information. If we talk about the type of genome, so there is two broad category of genome. Many of the people nowadays working on the prokaryotic genome and now the eukaryotic genome. In the prokaryotic genome, you know that they are very simple, and their genome size is really very small, you can easily analyze. But in the eukaryotic genome, we are dealing mainly with the human genome, plant and livestock genomes. So in this, what the other genomic information contain other than the nuclear is the mitochondria and chloroplast for the plant, and for the human and livestock we are dealing with the nuclear and mitochondrial genome. So if we talk about the mitochondrial and chloroplast genome, so why these genomes are essential? Because these are generally inherit from the maternal side, and this provide information related to the evolutionary aspect. If somebody really wants to look this particular species, from where it is originated, what are the origin of this particular species, any particular species, for that we extract the mitochondrial genome or chloroplast genome in the plants and study the evolutionary aspects. You can find many papers related to this. So this particular genomes are really useful to study the evolutionary biology or molecular evolution. And the size of this mitochondrial and chloroplast genome is actually it's a circular, right? The nuclear genome is generally linear, which pertain all the information etgc. Now just go to the subfields of genomics. So in genomics, it's broadly characterized into four different types, but now we are having different other genomics like epigenomics and single cell genomics are also now emerging field. So in the genomics, if we talk about, the first one is the comparative genomics. This is known as synthetic genomic, in which we are comparing one species with the other species and seeing what are the different variants present in one species makes different species unique. The second one is your mutational genomics, which generally give you the information about the mutation. For instance, a single nucleotide polymorphism and their variations, including indels and all those things. What are the mutations arises during the course of evolution? So this kind of studies is belongs to the mutational genomics. Then functional genomics, in which we generally deal with the functional validation of that particular gene, and this generally we perform with the cloning and all those CRISPR thing. And the structural genomics, it's pertain to the structural information with that particular gene and how structurally they are close together and diverse together. So all these information, yeah, we can fetch through the structural genomics aspect. So my lab is mainly working on all the four different domains of genomics. So we are looking in the all these four different areas of genomics. So now we talk about the advancement in the plant genomics. So I will touch mainly on the agriculture sides. What are the recent advances has been already made in this particular area? Also, like what are the future trends going on? So the plant, in case of plants, the first crop has already been sequenced was Arabidopsis thaliana in year 2000, and this is actually very small genome. It has 125 MB and total 25,000 genes present in this particular crop, and this is also known as a model crop, like in C elegans. is a model organism. Similarly, in case of plant, Arabidopsis, why we choose Arabidopsis first? Because this is a model crop plant in which everybody wants to investigate the study. And later half a decade we have the first serial genome, that is rice, and it is considered as a gold standard genome sequence. And we proudly can say that India is a part of this genome sequencing project. And because of this, we can able to venture into the genomics area. And NIPB, National Institute for Plant Biotechnology, IRI New Delhi, and DU South Campus, they both join hand together, and they are the part of this genome sequencing of rice, and they sequence together chromosome 11. So that time what happened, everybody has to sequence one chromosome. So every country has distributed the chromosome wise distribution, and you can see that our flag is right here, the chromosome 11, chromosome 11, and India sequence chromosome 11. And similarly other countries have contributed on different chromosome. Earlier it was whole taken care by the Japan and China. The people are involved mainly for this genome sequencing project, because they think that that time we are not having that much of facility, also not that much of skilled manpower to get the things done, the sequencing done. But we take this challenge and we able to successfully complete this genome sequencing project. So this was the first genome sequencing project in which India has contributed substantially for the chromosome 11, and it has been published in Nature. And this is considered as a gold standard because that time it was done by the back cloning method. Okay, back cloning. And the genome size was 389 MB, and total 36,000 genomes. So 36,000 genes, protein coding genes are present in the rice. Then after almost half a decade, we have completely indigenously sequenced this pigeon pea genome sequence. This is Arhar, and the two group from India, one from the ICRISAT and other from the NIPB New Delhi, Professor Singh. They sequence these particular crop. Why this crop? Because this is native to India, and also it is very interesting to know that this particular crop is grown in Gujarat also. So it is one of the most important crop for Gujarat. So it has been sequenced in 2011 and published in 2012, and its genome is 852 MB and total 47,000 genes present. But still, this is in in a not in a gold standard form. It's in a draft form. So we need to actually make a gapless genome of pigeon pea. So currently, I'm working on the pigeon pea genome sequencing, in which we are looking for the gapless genome assembly of pigeon pea, and we will soon publish this particular. And also, I was a part of Mango Genome Project with ICAR and IPB New Delhi Jute. We have already published from India and tea, in which we developed first haplotype map of tea. So nowadays, more than 1,000 total plant genome has already been sequenced, and day by day we are sequencing due to advancement of technology. Because earlier it was the first generation sequencing, now we are having the third to fourth generation sequencing platform available, through which we can take the benefit of this sequencing technology, and we can able to produce more gapless assembly, which is a kind of a not only gold platinum standard genome sequencing. So
Madam,
yes.
why there is gap in all genome sequencing? What are the reasons? Can you briefly share?
Yeah. Yeah, so why the gaps pertain in the sequencing? When you do the sequencing, so that time we were doing in the short read sequencing, right? So वो एक बहुत fractions में होता था, so gap was there. So वो small contigs. Initially we have contigs. I'll discuss you later. So वो contig होता था that we need to join to make a scaffold. So when you are joining, so the joining led some of the gaps because it was not fully sequenced. We can say that sometime it has having some handling problem also software was involved in that, because everybody is not aware how to and for which platform you have to use which software. That I'll come later. So these are
एनजीएस में तो नहीं होता है गैप? एनजीएस में होता है गैप?
now this like you can say अभी पैक बायो रिव्यू 2 आया है या फिर पैक बायो सीक्वल 2 आया है. इफ यू आर डूइंग हाइब्रिड जीनोम सीक्वेंसिंग उस केस में नहीं होता. इफ यू आर डूइंग लॉन्ग रीड सीक्वेंसिंग एंड ज्वाइन विद द शॉर्ट रीड, जैसे गैप जहाँ पे है उसको आपने शॉर्ट रीड से मैप कर दिया, सो देन इट विल बिकम अ गैपलेस. इफ यू सी द एग्जांपल ऑफ ह्यूमन जीनोम सीक्वेंसिंग, वो कितने पहले पब्लिश हुआ था. नाउ
Yes.
2022 we are having gapless genome sequence of human, right? Why it happened? Because earlier version of human genome was having gap, but due to advancement of technology you can have a gapless genome sequencing. जब gapless होता है तो आपका जो gene है वो भी that also get reduced, because then you have to do re-annotation, right? So this is all the iterative process. Will come later on in my slides. I have already
Okay, madam.
explained that.
Thank you.
Yeah, so now we are having more than 1000 plant genome already sequenced, and also many of the has already been involved in the Darwin tree of life or earth biogenome sequencing project, in which we are sequencing all the species present in that. So if we talk about the evolution of sequencing technology, so as I mentioned, the first generation sequencing, in which we are having the Sanger sequencing, Maxim Gilbert, we generally read in our literature. So these are the in which the earlier on the basis of first generation sequencing, human genome has been sequenced, and also the influenza was sequenced in the first generation sequencing, even in Arabidopsis also. Then after that second generation sequencing came, we worked on second generation as well, जिसमें 454 था, जिसपे राइस जीनोम सीक्वेंस हुआ था. So Solex, Iron Torrent, and Illumina. So if you have to do amplicon sequencing, in that Iron Torrent is really good. Small gene का जो sequence करते हैं. So what is the difference? Main difference is in terms of yield. You can see that इसमें 500 to 1000, इसमें 5 approx 500 base pair of fragments you are getting. But in the third generation sequencing for say PacBio, Oxford Nanopore sequencing native real time DNA, and you can have data in the KBs, not in base pair, you are having in KBs fragment. So आपको एक जो बड़ा length you are getting, right? So that is the beauty of the long read sequencing. But still people claim that the long read sequencing is not good because they are having lot of gaps. Why these gaps persist? Because in this platform people are generating the platform, but they at the same time they have to make the good software to analyze those high and data, right? To cater those data. So that is one paucity in this particular area. So that for this kind of system we need to have a good software system support also to analyze these kind of data. So now for example in PacBio, people are having their own analysis platforms in which you can analyze the data for NGS. So they are making that. So similarly, these are the some sequencing platform in the different chemistry. I'll not go more on that because you have already been covered. So what is NGS? Because we are having several different level of sequencing, so the next generation sequencing considered as a powerful platform. Why? Because it can generate millions of reads in a single SMRT cell. एक cell से वो बहुत सारे reads को generate करता है. So that is one beauty of that, and it can produce a high throughput sequencing data, because now the era of having high data science, everybody wants to have a good data point so that they can analyze their things. And also with the NGS we are having जितना ज़्यादा sample होगा you have a reduction in cost. So that is also one beauty with the NGS. So and at a time you can sequence many samples also in a flow cells like that. So all these benefits we are having with this NGS platform. So if we go with the generation wise, so first generation was the Sanger, then second generation was the pyro sequencing and sequencing by ligation. Third generation is SMRT, the PacBio system, semiconductor nanopore sequencing, and the fourth generation is single cell, in which we are just doing the single cell wise analysis. These are the some pictures of sequencing platform. For example, you can see the Nova 6600. This particular platform is already in at GBRC. Nanopore Promethean. This is mainly useful for the higher organism, eukaryotic, especially for example for the plant people, those who are working on the longer genomes, because for example tea, sugarcane, wheat genome. So these are the good, because in the nanopore also we have a nanopore, promethane, methane, mini ion. So different platforms are available. So according to your need, you have to choose the platform, and then you have to run. And now the most latest one is PacBio SQL 2, 2E, and Review. Now they have the one advanced system which the foreign people are working on, the Review 2 system, in which mainly useful for the long read sequencing, and they have a good data quality. Also, they have their customized software in which you have the sequencing data. You can do the this assembly and ready to go. So it is one chromosome, one sequence file, like that. So chromosome wise data you can have with this good platform. So, if I talk about the revolution in the arena of genomics, so it is actually good thing that we are having lots of sequencing platform. Everything is there, but what is the major challenging aspect is the compute omics, because you have lots and lots of data, but where to store, right? कहाँ पे आप रखेंगे, and how you will analyze? You cannot do such type of analysis on your own a laptop or in-house small system. For that you need to have a good computational power, computational sources, so that you can analyze the data. Also the good server system, so that you can analyze the data, because once you start analyzing, intermediate files made, and they acquire lots and lots of space. So for bigger genome you need to have a good server support and server system. Now come to the assembly. Different types of assemblies are there according to the platform. So first क्या होता है, when we send our sample for the any sequencing, according to our objective, like what you want to develop a reference genome. For that you have choose the platform accordingly. If you want to do resequencing, for that you have to choose the platform. And if according to your study, if you want to do transcriptomic, accordingly you will choose, and accordingly you will choose the coverage. But after that, what we have to perform the assembly. So there is a two different ways to do assembly. One is for the long read, and the other one is for the short read. Short read is your generally Elemina platform. So short read Kamer method पे based होता है, and इसके different softwares हैं, right? And long read is your based on the overlap layout consensus sequence. So for that the software is Flies and Falson. And for short read you are having spade a bias, like that. So what is the major difference between these? This is actually based on the Kamer method. So short read one, so you can see that this is a 7 mer, and sliding window is going on, and that's how this is the deep Brueggen graph method. And likewise you can see the nucleotide bases where the joining together. And in this is actually based on the overlapping read 1 to read 2, and जिसका सबसे ज़्यादा डेप्थ होगा, and accordingly they will call. So that is the assembly. You can see here read 1 and read 2. So this read 1 and read 2 joined together, we have the assembled one in long read, right? So you can see it very clearly. So ये यहाँ से कुछ भी नहीं छूटा. We have the two overlaps of this red segment, and we can able to catch the yellow, red, and blue, right? All of them. So that is the for the long read. And if you are having the nanopod data and PacBio data, for that you have to use Kanu Flies, Falcon Unzip. And for the short read you have to use Speed Bias, these things. So this is the overall platform of the sequencing data analysis. Like how Dr. Jha, you were asking na ki why the gaps are there. So here is your answer, that first it is kind of jigsaw puzzle. So first you have a raw sequence read. After sequencing you have a data in a raw sequence read format. So you can see that your data is in this format, the small structures are there. So this is the
हेलो, आपकी आवाज नहीं आ रही है? एम आई ऑडिबल?
is to assemble this contig. So this is known as contigging. Assembling read into longer pieces called contigging. So in this, what we will do, we are just joining this small contigs to contiggings. So these are the your big chunks. And after that, this contigging, you have the scaffold. अब ये जो है इसके बाद you have to make a big chunk is known as scaffold. After scaffold you can see that these two are joined together, but they can join only if we will place n. So this n is known as gap, right? There is 4 gap in this particular sequence, and these are all your nucleotide. blue color is your nucleotide. So why this gap is there? Because we just wanted to join, but they will join only if we will insert the gap. But we can remove these gaps through polishing. So for polishing, we are having different tools known as Racon and Pylon. For this, we can polish this particular scaffold, and then we have a final scaffold. And then after final scaffold, we will have a pseudomolecule, means chromosome level of assembly. So that is the whole part of the assembly. So assembly is also split into these part. First one is small contigs are there, then you have to perform the contigging, then you have to make the scaffold. After making the scaffold, you have to perform the polishing, because there is a gap present in that. We want to remove the gap. पॉलिशिंग में हम क्या यूज करेंगे? वी विल यूज द स्मॉल रीड सीक्वेंसेस. फॉर इंस्टेंसेस इलिमिना का जो हमने स्मॉल रीड किया है, दीज आर योर लॉन्ग रीड. सो स्मॉल रीड को वहां पे अलाइन कराएंगे. सो आफ्टर दैट वी विल हैव टू द असाइन this nucleotide, and then you have a full stretch of gapless scaffold, and then that scaffold leads to a pseudomolecule, means chromosome level of assembly through hi-fi and other platforms are there. Now come to the sequence processing tools and applications. So that was the part of the assembly. Now this is the part of the processing and their application. So these are the different sequencing platforms. You can use any of them, like Illumina, MySeq, Ion Torrent, PacBio, like that. Then you have a sequence read. After sequencing read, you have to do the quality assessment ki jo sequence aaya hai, whether it's good or bad. So we have a fast QC analysis for that, in which if your Q score is Q30, if your Q score is more than 30, then it is good quality. If it is a below 30, it is a bad quality. So after that, there is a two-way. First one is your de novo assembly. De novo ka matlab hota hai starting from the scratch, and the second one is your the reference based assembly. De novo means you are doing very first time. There is no any previous assembly or sequence reported, but reference one is actually there is already one reference present, and you want to based on the reference you can do the alignment and assembly. So for the de novo is one method in which we can do that, and for the reference based, in which we are having already the reference present. And in the both the cases, we have to perform the same ordering of contigs, quality assessment through BOSCO and cost analysis, and then we go for the genome annotation. Genome annotation is assigning the genes with a function, and for this there is a tool called blast2go, and this NSassemble, rust. So different tools are there for assigning the genes ka function. So this is the complete sequencing process. Is there any doubt in this particular process? Because it's look little bit complicated, but it is not that complicated. With the left hand side, I have written the software name. For example, if you can do the alignment with BWA, BWMM, Bauti, CLC Genomics, like that, and same de novo assembly के लिए Velvet Soap, Mega Hit use करते हैं. Likewise for the assembly, you can use the different tools for reference based as well. Should I move forward or?
Yes, yes. पूछना होगा पूछ लेंगे.
Okay. So
Yes.
now this is one report of fast QC, in which you can see that there is a clear cut distinguish. This is the left side is the good quality, and right side is one the bad quality according to my screen. You can see according to your screen as well. So you can see that यहाँ पे the first one we are having all the Q30 more than Q square is more than 30. You can see this one, but here you can see that it drops drastically. It goes up to 10 even. So this is actually your bad quality. So this is your the quality assessment of the sequencing. You have to check the quality whether it's good or bad, and then we go for further assembly process. So this is the first step in the sequence data analysis. Now this is already I have explained. This is the genome. First we got the reads, and read we are doing contigging, and contigging, after contigging we are making the scaffold, and these are based on the Kmer method if we are using the short read sequencing. If we are using the long read sequencing, in that case we are using the overlapping scenario, especially for the Kanu flies. These are the software meant for long read sequencing. So yeah, same. This is the same thing like the one contig. This is just for an example, and you can see that these are the ATGC pair and how they are aligning together. So these are the some de novo assembly and reference based assembly software. So assemblers are A5, Abias, CLC, Allure, Edina, Soap de novo, Velvet. So these are the some softwares names to do assembly, to perform assembly. So accordingly you can do that. Now come to the very interesting question, like how to estimate the genome size based on the K-mer distribution. So for example, if your sequencing data comprises 20 GB, ठीक है, 20 GB of data and the coverage is 10x, so your genome size would be, so this is a very simple method to calculate the genome size is equal to total base pair by the coverage. So total base pair we are having the 20, right? And coverage 10, so we are having total genome size is 2 GB. So that's how you can estimate the genome size of de novo species, right?
बट कवरेज कैसे निकालेंगे?
Okay, so coverage that time you have to say the coverage now. If you are doing, इसका भी बहुत एक interesting aspect है. If you are doing de novo 100x coverage we generally prefer, ठीक है? But if you are having already reference and you just want to do the resequencing, so 30 to 50x coverage is fine, just to mind the variance. So what is your objective? It's all depend upon your objective. कि आपको क्या आप what you want
साइज
to.
बाद में हम कैलकुलेट करते हैं सीक्वेंसिंग से पहले या बाद में?
Yes, बाद में.
ओके.
And once you have a data, because this is all based on the data, no? Yeah.
और कोई मेथड है जीनोम का साइज पता करने का या
That is,
सीक्वेंसिंग?
no sequencing. This can tell you roughly, but actual तो wet lab से करते हैं जो karyotyping और वो सब वाला जो method होता है, fish and all those things. So उससे exact पता चलता है chromosome number और उसका पूरा जो size है.
टोटल जीनोम साइज.
Total genome size तो आपको tentatively data से ही पता चलता है. For that you have to have sequencing data. Yeah, उससे आपको chromosome number पता चलता है karyotyping से.
ओके, ठीक है.
So now we have to do the assembly assessment. जैसे for example, if you have the already performed the assembly, वो क्या था, sequencing का assessment था Q30 वाला. And this is for the assembly assessment. You have done the assembly, whether it's a hybrid assembly or the single end assembly. For that you have two different software, ICARS and Quast analysis, as you can say that quality assessment tool for genome assembly. So in that you have some graphs which that graph can tell you based on the reference. If you have already reference available, तो उसके base पे it can plot कि आपका कितना coverage हुआ है. If it is not reference based de novo, then you have to take the sum of the related species to perform the assessment. So that's why. And in ICARS also it will show you all the assembly parameter along with the kitne gene cell length hai, all these for the assessment that your assembly is of good quality or not. If not, so you might have to change certain parameter, or maybe you have to choose some different software which is suited for your platform, because as I mentioned, for the your previous paid method won't work. Okay, for that you have to go for the flies, because flies meant for your nanopore sequencing. So you have to choose that way. Sometime kya hota hai, what is the problem happening, people are using long read sequencing platform, latest cutting edge technology, but for the analysis they are using the old softwares in which they are comfortable. So that's one thing which we should avoid always. So first you have to read thoroughly according to your data which software, which tool is useful, and how you can use and get meaningful information out of it. So that is the key consideration which we have to take care while doing the assembly or any sort of bioinformatics analysis. Now come to the annotation part, genome annotation. So as I have already mentioned, the genome annotation is a process of attaching biological information to the sequence. We have the sequence, but we don't know that particular sequence or that particular gene, what it does, what is the function of that gene. So that we need to identify. आपको पता होता है, for instances, in case of rice we have drab gene. Drab is responsible for the drought, right? So how you will identify this particular sequence belongs to DREP gene, this particular sequence belongs to RAP gene, this particular sequence belongs to DELA protein, this particular gene belongs to dwarfism in plant. So how that information we can gather, so that for that the process is genome annotation, so in which we are attaching the biological information. So it consists of two main steps. First one is identifying the एलिमेंट्स और जीनोम प्रोसेस फॉर द जीन प्रिडिक्शन, जिसमें दो फाइल बनती है, GFF and GTF files are there. And then after these we are attaching the biological information. First we are predicting the gene, that how many genes are present in the genome. And for these there is a tools like a custis is there. And after predicting the total number of genes, what this particular base pair particular genes are performing to, what are these particular genes encode for that particular function. So for that there is a functional annotation. So annotation is basically two type, one is structural annotation, the other one is functional annotation. So accordingly we have to perform, and for these there are the different tools and softwares are available. So you can see that for the structural annotation we can identify the open reading frame and their localization, gene structure, coding, reason, location of the regulatory motive, and these are the certain pipelines. And for functional annotations, we can identify the biological function, biochemical function, expression involved in regulation and interaction of the genes. Now this is one interesting software, that is IGV. So in IGV we are generally using the this your integrative approaches of, like, we are just seeing the genomic data in terms of number, but if I really want to look particular segment of gene or particular segment of inversion deletion present in the genome, for this we are using the IGV. So it is freely available, this particular tool, anybody can use it. So that's how the IVG looks like. If you just zoom out, this particular line, red, green, blue, that is actually encoding for the nucleotide basis. So if you zoom out, so all these nucleotide you can able to see with your eyes, and you can see the difference with one individual to other individual. So you can simply look at the absence and presence of genes in that genome, because you can see these are the different BAM files are there. Now come to the, so that's all the parts of the genome sequencing project, but this is the concise view of the goals of genomics, in which first we have to decide which plants we want to sequence, whether we want to do the simple gold standard reference genome, or we want to perform the pen genome. Pen genome mein you have a multiple genotypes, diverse genotype of single species, single plant, and in which we are performing the structural variant analysis. So first you have to choose the plant for genome sequencing. After sequencing, you have to choose the platform, which platform is suitable for your analysis, and then after that sequencing data we have to assemble the genome. After assembling and or making all the pseudomolecule gene prediction, everything ready to go, we have to perform the annotation through blast to go. In annotation we are having two different types of annotation, one is structural annotation, the other one is functional annotation. And we are having used data set, right? So where to store this particular information, so that for that we are developing the genomic data bases. For instance, in case of rice, there are 3-4 different databases. Oriza DB is there. Okay, so Oriza Origin database is there. Oriza SNP database is there, along with the NCBI. NCBI is the hub in which we are just submitting the data, but there is a species specific database are also there for their to store their all genomic information. After that, you can pinpoint, because you don't want to look for the entire genome, you're not a genomics person, you just wanted to work on a small segment of the gene. So you can identify from that data set the small genes of your interest. For example, you are working on the biotic stress, somebody is working on the abiotic stress, somebody is working on the nutritional aspect. So through these huge information which somebody has collected and deposited in the databases, you can mine your genes and do the structure variant analysis, and further you can identify the potential candidate genes which can go for the gene and genome editing to develop superior crop variety. That is the climate resilient crop variety, we can say that, which is comprising the nutrition dense attribute. So these are all the entire goals, like why we do the sequencing. So we have started from scratch, from the sequencing to the gene editing. Okay, transgenic, or we will not say the transgenic actually. Yes, so we ended up with the development of superior variety which can benefit the farmers community, also the consumer, right? So this is the overall goal of plant genomics. So now in this line, you can see this is very recent project launching, and globally people are working on that, the Earth Biogenome project. So and also the other project, and this is the UK based project, is the Darwin Tree of Life. So they want to sequence each and every individual present in the Earth and identifying how they have ancestrally close together and diverse together at one point of time. So they, why they wanted to do so, they have answered the question that why they want to sequence life to revolutionize our understanding of biology and evolution. The first aspect of this project was, and the second is conserve, protect, and restore our biodiversity. And third one is to create new benefit variety for the societal and human well-being. So that is the whole theme of this Earth Biogenome project, in which India is also part of this particular genome sequencing project. Now come to the other aspect of genomics, that was the single crop single thing genomics, the pen genome. As I have mentioned, that pen means you can have a lot of diverse species of same diverse genotype of same species, that is known as pangenomics. So pangenomics combine the genetic information of multiple genome to create a comprehensive reference genome to capture the genomic diversity or genetic diversity present in that particular species. You can see that ki ek particular, for example rice here, so different types of rices are there. You know that we have basmati, we have small grain rice, we have bold rice, we have starch resistance rice, we have black rice, wild rice, different types of rice are there, but they all belongs to rice or rice. So what are the different genes in which one particular variety is more tolerant to other? So what are the diversity existing in that? So that study is can be done through the pangenomics. So it was started in 2005, and now it's keep growing on. You can see these are the different crop varieties which we have done. Initially it was started with the rice, and now you can see not only in the crops, also in the horticultural crops, fruits, different tomato, tobacco, legumes, everything every peoples are doing on the pangenome thing. And you can see that these are the some database in which we have all the data available for that, that is plant pen database. And yeah, this one I wanted to highlight, that IBDC. Now earlier we have NCBI
Hello.
for. Hello.
Hi, ma'am. I wanted to ask that if we are searching a particular protein for a particular plant,
Hmm.
we can't get much details and research papers available. And if we are targeting a particular protein, how can we get collect data, all the data we have required?
Varad
So you know,
Patil ji, please introduce and then ask question. That will be better because at which standard you are accordingly answer may be Hana, modified or can be discussed.
Okay,
Please.
okay. Sorry, sir. Sorry, sir.
हाँ, बोलिए.
hmm.
Myself, Varat Patil, and I am from IR Institute of Advanced Research, and I am doing MSc Biotechnology, and I am particularly in second semester now.
Okay.
Okay. So you are looking for a particular set of protein, right? And you want to fetch the all related protein of in different species, right? That is
Yes.
your question. So for that, for protein, actually, you which database you are using to fetch the protein sequence? Which database, or it's your own generated sequence? Like how you get those sequence, protein sequence?
Uh, Particularly yeah, जो EMBL plants वाला डाटा है ना, EMBL plants, I was using that.
So do you know Uniprot?
Yes, yes, I know.
So in UniProt, for example, if you have X species of that particular protein and you want to look for the entire other species, so simply you can type that protein name and you will get list of information related to that particular protein. For example, if I wanted to search for Della protein, okay, Della
Okay.
protein is responsible for seed development in plant. So I will just simply write della protein, and it will give just maybe just be species में della protein present होगा, वो सारा का list दे देगा, and you can download, uh, you can go to the download and you can download in whole data at one time. That file comes in a zip.
Sometimes there is not all the data available. Like I also go through research papers, but there
So
are
after
also lots of
lecture,
things missing.
Bharatji, so after lecture, na, we can discuss long discussion,
Okay,
है ना?
okay, okay,
That
okay,
will
okay,
be better. So
okay,
wait.
okay,
Yeah,
okay.
I can answer right now. If all the data is not available, it means that person has worked, published, but not submitted to the database. In that case, we can't, means you cannot fetch for the information. So in that case, you have to rely on the literature also, if something new, because that's why these databases have been created now, so the people can do the sequence or predict the protein or peptide. So they have to submit those information, then only you can able to track. And if you go dig deeper into that, you will find which paper it is that sequence has been deposited, so you can find the literature also. But if somebody has not submitted in the databases, so how you will sure that it is authentic or not? That is also
Yes,
one
yes.
now. So
Got,
better
got it, got
always,
it.
yeah, so better always go with the authentic source
Okay, okay.
than.
Thank you, ma'am. Thank you.
Yeah. So I just wanted to highlight the IBDC. This is recent launch from the Indian government. DBT has launched this, and abhi everybody has heard about the 1,000 genome sequence project, 10,000 genome sequence project of human. No? So Modi ji has released that data, and GBRC was also the part of that genome sequencing, human genome ka tha. So that all data has been deposited to the IDBC. Now it is, being an Indian citizen, an Indian scientist, it is it is mandatory to submit the any sequence data, whether it's a gene sequence, genome sequence, or protein sequence. If you have sequence from somewhere, you have to submit to the IBDC. If you are submitting to the NCBI but not submitting to the IBDC, it will not get the recognized. So first you have to submit to the IBDC, then you can submit to the elsewhere any databases. So this is now mandatory guidelines, right? So it's a very big data center at Thisti, Gurgaon, IBDC, जो DPT का part है, and they have made it. And Dr. Saurav Raghuvanshi is the leading scientist who headed this database. So you can see that Indian Biological Data Center, and it's Indian Biological Data base in which high throughput phenotyping data they have created. Different types of databases are there. So in phenotyping, they have only kept phenotypic information. So likewise, so you can see that Genome Med India project. So all the data of genomics is submitted in this particular database. So now those who are working on the sequencing aspect, they have to submit the data to IBDC. So it is compulsory now. Now come to the tools development. So from my group, we have developed one tool that is useful for the hybrid genome assembly, right? So we are doing actually for PGNP or her variety, we are doing the sequencing for the nanopore as well as Illumina short read sequencing. But that time we are having the trouble like how you will analyze, because these software are meant for only for one platform. Nobody that time was doing the hybrid genome sequencing assembly. So we developed the genome assembly software pipeline using platform Python and Docker, so we can able to do the haplotype based assembly and develop the high density linkage map. So these are the some stats. I'll not go much detail into that, but I'm just telling ki if you have a data, so it's not always necessary ki jo existing tool hai that will suffice your need. Sometimes you have to create your own data. You have to create your own tool to analyze your data set and also cross check it with the quality assessment and other parameters. So for these hybrid genome assembly we developed, and that can useful for the nanopore sequencing and Illumina sequencing. We have not we can try for the PacBio as well, but that time we did with the nanopore and Illumina hybrid genome sequencing. So we actually assembled total 10 different genomes of PGNP. Now this is one interesting project in which we are working currently. So this is based on the triose binning assembly. So you know the heterosis now, everybody knows heterosis, hybrid vigor कहते हैं जिसे. So this is actually the work of Mango from the ICR. I am just showing off one fraction of this work in which I was involved in. So there is a two variety of mango. One is Neelam, the other one is the Sherry. So the Sherry is the maternal and Neelam is the paternal. And you can see the total number of gene. After sequencing, we sequence through the hi-fi PacBio and with the 300x coverage, assemble both maternal and paternal genome. And actually, the Amrapali variety is nothing, it's just a cross of Neelam and Dasheri. Okay, so you can see that how the heterosis is actually proven here. So total number of genes in Neelam is 36,143, and in the Shari we are having 36,273, and unique gene to this particular Neelam is 9,614 we have identified, and here you can see that 9,747. So if we find out the common gene present both Neelam and Dashari is 26,529. Rest are the unique to Dashari and unique to Neelam. And this is actually Amrapali is a hybrid of Neelam and Dashari, and we identify that it has more, almost 9,000 more number of gene than Neelam to Dashari. It has total 45,890 gene. So it shows that this particular variety Amrapali is superior than Neelam and Dashari because it has a gain of gene. So there is a two type of concept. One is loss of gene, the other one is gain of gene. So the gain of gene is means your heterosis. Loss of gene is your inbreeding depression, right? So you can see that through bioinformatics, through data, through sequencing, we can able to analyze this type of study. So we are working on this. This is one actually story which I just wanted to share, like what you can do with the sequencing data and how it is benefiting in the horticultural crop as well. So this is one segment. So this type of study generally we do for the livestock genomics, in which we have parent 1, parent 2, and we want to study the offsprings, right? So trios binning sequencing उसे कहते हैं. So that is generally useful for the your large animals like cow, buffalo, or there is a couple of paper, but this particular study we are reporting very first time in Mango. Now we have do the mango genome sequencing, and through different varieties of mango we are able to develop the 82K SNP genotyping chip and useful for the diversity analysis. So this is the small fraction of study in which it is published, but we have done larger scale of study also. Total 387 diverse genotype, but this particular study is based on 84 diverse mango cultivar, in which they are stratified into 7 different groups, and you can see that, and this actually belongs to the region wise, and also perform the genome wide association analysis of agronomically important trait, and the phenotyping data was collected by the IARI and IHR Bangalore, and we performed the genotyping because we have the gene title. So we did genotyping and perform the genome wide association analysis. So out of 80,000 marker, you can see that these are the above threshold, these are the some strong association, and these small individuals are actually your genes. So these are the strongly associated genes that fruit length. Similarly, these are the at chromosome 4, these are the strongly associated gene related to the food weight, polyembryony, pulp percentage, total soluble starch synthesis. So that's how you can mine the important genes. So this is kind of mining, वो सागर में मोती खोजने की तरह है. So you are mining very useful genes which out of 80,000 marker, out of 20,000 genes, you are mining one or two genes which are strongly associated with that particular trait. And in the other study, so this is actually why I'm showing here. So this is the first report in India. We have published that in Bioinformatics Journal, and this is the first haplotype map of T. So earlier the haplotype maps were developed for maize and human genome, but none of the attempts has been made through from India. So this is the first report from the India side, and that I developed whole pipeline. So this is the complete pipeline, and the chromosome we made through software, in-house developed software, not the freely available or some of the access software, and also developed the T-HapMap database, in which all the, as I was mentioning, that now we have a dedicated database for the species. So this is the dedicated database for the tea. Tea, in which we have all the information, all the genotype, all the sequencing data related to the tea. So this is the highly appreciated paper through from the ICR, and this particular haplotype has been generated through total 369 diverse set of tea genotypes. So you can imagine, and one single data we are having 4 GB of genome. So T has actually 4 GB of genome size. So you can see the data points we have handled through this, through the large system. Now come to the SNP genotyping platform. So these are all the sequencing part. Now I will talk more about the genotyping platform. So since sequencing, we can mine the variant, we can mine the SNP. SNP is single nucleotide polymorphism. Every individual is 99.9% similar. 0.1% variation is because of the SNP, and that makes one individual unique to other. So this is the overall concept of the SNP genotyping assay. So there is a two different methods of genotyping assay. One is low multiplexing with high sample throughput. Other one is high multiplexing with low sample throughput. So low multiplexing with high sample throughput useful for the population structure study, foreground selection, diagnostic purpose, seed quality control. And high multiplexing with low sample throughput useful for the evolutionary study, association mapping, background selection, genomic selection, and diversity analysis. So these are the varied exam applications of different genotyping platform. So we have developed, so this is the work from my PhD part, and also some of the recent funding from government of India, DST. So we develop high throughput SNP genotyping platform for rice and pigeon pea. So I did my PhD on rice. So this is the Affymetrix platform, and you can see that we have a patent of this particular genotyping chip. And because of this chip, it was deduced that Chinese people always claim that rice did originated from China, but because of this chip, we are able to study the evolutionary aspect, and we claim that the cultivated rice, which like Oriza, Rufi Pagon, Nivara, and Sativa, is actually originated from the India. It's not coming came from the China. So we claim that, and we have the patent of this particular work. So we developed 50K rice SNP genotyping chip, Pigeon P62K SNP genotyping chip on the Gene Titan platform, and now developed Mango 80K SNP genotyping chip. So how genomics help in crop improvement? We know that all these technology are there. So for what is the application of that? So first one is we can harness the genetic information present in that. You can see that this is a beautiful example of maize. So you can see the maize, Baba McClinton worked on that jumping genes, and you can see that the corns, they are having beautiful color. On a single corn you can see different kinds of colors are there. Why this happen? Because there is a variation present in that, and that variation we can deduce through the genomic information, through the sequencing information within the species, means within the maize, also across the species. You can see the across the species, like different species you can use, like monocot, dicot. Also you can take pseudo cereals. So across the species, what are the varied variations existing? So that also we can harness through the genomics. We can do the population structure analysis. So we have actually performed this particular study, collected total 418 diverse Indian wildlife succession from different ecogeographical region of India, and based on the genetic information, we have stratified into that which one are the pure type, which one are the admixture type. So population structure analysis we have performed, and how much the percentage is actually crossed between the species, different species, and make a new species. So you can study the diversity also. We can trace the history of crop origin and domestication and their relationship with the wild ancestor. So because due
और
to
कितना स्लाइड है मैडम?
um 5 or 10 होगा, 5 I guess. I will be
ठीक है,
quick.
ठीक है, ठीक है, ठीक है मैम.
So we can able to trace the domestication study with this. Now the whole genome association analysis, which I have already discussed, the genome wide association analysis, in which we are currently working on one project, that is PGNP project. So in which we perform the genotyping and phenotyping of the key important trait, for example seed coat color, total protein content, and amino acid estimation, resistance starch. So, and use the genome wide association statistical models and publish very good papers on this work. So you can see that also we have identified some of the key genes and reported some of the developed variety to the NBPGR. Also, you can identify the gene-based haplotype estimation, and this is also a first report from the Indian side to identify what are the genes inherited from the one genome to the other genome, that is known as the haplotype. And also you can identify the variety identification and seed purity through this genotyping technology. So you can do useful for the seed testing and all. And this is the one important study we are currently working on PGNP to mine the variation present on the seed quality and nutritional trait. And also we have developed the PGNP omics database in which all the information related to the PGNP is available, starting from the sequencing to the metabolomics aspect. So all comprehensive information is present in this PPomics databases. So these are the some important results which we have got through the genome-wide association analysis. Now come to the high throughput phenotyping. That was the genotyping part. Now everybody is looking for the phenotyping, like how you will do the phenotyping. So for that, we are actually having the robots and drones in the field to capture the data. So we are doing the high throughput genotyping, but now the era is also to capture, especially in the agriculture sector, to the high throughput phenotyping data. And these are the some different high throughput phenotyping data sets. So what are the different kinds of advantages of having high throughput phenotyping? First one is it facilitate the rapid non destructive, because human made nahi hai to error nahi hoga. It can give you high throughput detection. Also it can address the traditional method, which is generally based on the observation, right? So this is actually useful for the real time monitoring. Large scale plant traits can be measured at a time, and also you can discriminate through the high throughput genotyping different cultivars, and at the same time we can predict the yield of the crop and selecting genotype trait of interest. So it make actually our life more easier as compared to manual thing. So that is the beauty of this high throughput phenotyping platform. Now this is the era of multiomics. Not only the high throughput genotyping, phenotyping, we need to have the high throughput metabolomics, transcriptomics, proteomics, ionomics. So these are the all integrated omics platform that is known as multiomics approaches. Now people are looking for the multiomics approaches to predict one particular traits of interest. So these are the some couple of database which we have developed here at GBU, and it's working really well. For example, PPomics databases and LDDRG, legume disease resistance databases, in which we are having all the genomics information along with all the other trait related information which can useful for the agriculture sector. Now this is, I think, my last slide. So what are the actually benefits? So we are and started with the genome. From genome we mine the trait of interest of gene and employing the multi-omics approaches, and then we are employing that particular things to the field and what goes to the farmer. So actually now the study is, earlier it was one gene, one PhD student, like that. Now the one genome, one PhD student. So now the gene, and then we are doing the multi-omics approaches, and what these through these approaches we can plant it to the field, and then finally we are getting good quality of food in the consumer plate. So that is the whole chain of agriculture sector. And through this technology we can able to, through CRISPR and genome editing, we can able to develop the bacterial blight resistance, fragrance rice variety, high grain number, large grain size, powdery mildew resistance, purple tomato you are now seeing, which is rich in anthocyanin pigment. Similarly, high oilic content in soybean, canker resistance in your citrus fruit. So these are the some wonderful examples which we are seeing in coming days for the this the product of agriculture. So now my takeaway is, now no, not only one omics is suffice the needs. We have to look multi-dimensional aspect, and along with this we have to incorporate now AI and ML, especially to make our prediction more better, or to identify more better quality genes. So the strategy is actually to integrate the multi-omics approaches to identify, to dissect the complexity in the agronomical level and predict the yield. So that is the things we need to take care, and now everybody is doing that multiomics side of science. So with this, I would like to thank you all for your time,